Hello, everyone. Thank you for joining us for today's webinar. We've invited Sarepta Therapeutics to join us to provide data updates from their first 3 studies with SRP-9001, their investigational microdystrophin gene therapy product. I'm Eric Camino, PPMD's Vice President of Research and Clinical Innovation, and with me is Pat Furlong, PPMD's founder and CEO. Joining us for this webinar are Stephanie Mason, Medical Director at Sarepta, and Louise Rodino-Klapek, Executive Vice President and Chief Scientific Officer at Sarepta. Just a couple of housekeeping notes before we get started with today's webinar. This is being recorded and will be posted to PPMD's website, so please feel free to share with any friends or family in the community who may have missed the webinar. We'll also be taking questions at the end. So as we go through the presentation, please feel free to submit any questions into the box at the bottom of your screen, and we'll try to get to as many of the questions as we can during the Q&A session. And with that, I think we're ready to start, so I'll hand it over to Louise to start the presentation.
Thank you, Eric, and thank you to Pat and PPMD for having us present the status update of our SRP-9001 gene therapy program today. And thank you to all of you on the phone, on the web for joining us today. We really appreciate it. It's with my great pleasure that I get to introduce our speaker, who is Dr. Stephanie Mason. She's a medical director here at Sarepta, and she's also the clinical lead for our SRP-9001 program. And she'll be taking you through an update of that program today. And so with that, I'm going to turn it directly over to Stephanie. Stephanie?
Thank you, Louise, and I'll just echo Louise's thank you to PPMD and to everyone in attendance today for this opportunity to chat with you about our results to date. These are our forward-looking statements. I won't read the whole slide to you as it's quite lengthy, but what it says is that we're going to talk about our hopes and our goals for the future. And because we're discussing the future, there are risks that may lead our hopes or plans to change. So today, specifically, we will be talking about delandistrogene moxeparvovec, also known as SRP-9001. I would also like to say that this product is in development, meaning that it's investigational and it has not been approved by any regulators, such as the FDA. Sarepta itself has a robust pipeline of therapies in development for Duchenne muscular dystrophy. Some are commercially available, such as the RNA-based therapies, also known as exon-skipping therapies, and others are in various stages of development. Today, as I said, we're going to focus on delandistrogene moxeparvovec, or SRP-9001, which is a gene transfer therapy. And I'd like to take a moment to just define what a gene transfer therapy is, in case there are people on this call that may have not heard that term before. A gene transfer therapy is a product where a modified virus delivers a piece of DNA into a patient's cells. Our product uses a virus called AAVrh74, which does not cause disease in humans. The DNA that it carries is a template to make a shortened form of the dystrophin protein. As I'm sure many people on this call are aware, the dystrophin gene in Duchenne muscular dystrophy is the one where the mutation is that causes problems with forming a dystrophin protein. So this template that gets brought to the cells has a version of a shortened form of dystrophin that is meant to take the place of the natural dystrophin. Our dystrophin has been carefully engineered in order to try and make it as efficacious or to work as best as possible when it's in the cells. In order to make this therapy as broadly available and as quickly as possible, Sarepta has formed a partnership with Roche, and this in particular enables a global scope for this product as well as potentially future products. So let's think about which studies that we're going to talk about today. Sarepta has several ongoing trials, and I won't discuss results from all of them, in part because not all of them have results available. The data today that I will be talking about comes from 3 trials that are in the box here shown. Those trials are known as 101, 102, and 103, and are marked as such. Specifically, I'm going to report today about 4-year data in the 101 study, focusing on the functional outcomes for these 4 patients. I'll also talk about the 103 study. The first cohort of that study, and there are several cohorts, but the first cohort recently reached their 1-year mark, and so we'll talk about the functional data at 1 year for those patients. We've also now integrated the results across the 3 studies in the rectangle there, 101, 102, and 103. And so when we group them together, we get a total patient number of 84. There are actually 85 in the 3 studies together, but at the time that we gathered this data, the 85th patient had not yet been dosed. So we're going to talk about 84 patients. So we'll talk about their safety data when we pool them all together, as well as the functional data for the ones who have reached at least 1 year. I won't cover the structure of each of those studies on this slide because we'll discuss them when we go to each study's results. I will note that the first 2 studies, 101 and 102, used preliminary or clinical research material, whereas 103, as well as the study 301 listed on this slide, use the same material that we would make commercially available when and if we're able to get successful commercial approval. Before we talk about the results of the studies, I want to spend a little bit of time discussing how we measure results in trials. As a patient, a parent, a friend, or a family member, the idea of how well a therapy works may be tied to how a person feels when they do their everyday activities, like walking around the block with your family, playing in the park, or moving around your house. But in a trial, we need to be able to put numbers and standards so that we can compare across people and across groups of people. So when we look in our trials, the first thing we look for is this box that's labeled in purple called expression. And expression is our way of talking about proof that the gene that we've transferred with this virus actually got to where we wanted it to go, that it was turned into a protein, and that the protein also got where we wanted it to go. These results have been shared before, and so I'm actually not going to talk about them today. And instead, I'm going to focus on the part that the arrow goes to, calling impact or function, as well as where it comes from, which is the safety box. So when we talk about functional outcomes, as I said, we need a way to compare across people. And so in order to do that, we use a functional score. And the score that Sarepta has used in its trials is called the North Star Ambulatory Assessment, or NSAA. Many of you may have heard of this score before or already be familiar with it, but it's a 17-item score that uses different activities such as ascending a step, descending a step, getting up off the floor, or running. Each of these items is scored with a 0, a 1, or a 2. A 0 means that the person cannot complete the action. A 1 means they can complete it with some form of modification, and a 2 means that they complete that action without any adjustment or modification. And what do I mean by a modification? Well, for example, when getting up off the floor, a patient with Duchenne may sometimes use their hands to push off of their own legs. That would be considered a modification of how to get up off the floor. And if they successfully stood up, they would score a 1 for having used their legs to help them. So now that we've covered some of these points about how we measure the outpoint of the trial, let's talk a little bit about how we compare between groups of people. Some trials are designed with placebos in them, meaning that there are people actually enrolled in the trial who don't get the drug. Our 102 trial, for example, had a placebo arm. But other trials give everyone the treatment, and those are sometimes called open label trials. And when everybody gets the treatment, you need something to compare against. One of the solutions that Sarepta has used is getting an external control set. External control means that these people are not in the study themselves. Their data is gathered through some other form, either in a different study or from, say, a registry or an observational database. So these patients didn't receive treatment, but they did get monitored for their functional scores and whatever endpoints the trial is using. And so we would use how those patients did over time in this external control cohort as the comparison or the stand-in for a placebo arm of the trial. As you can imagine, it's important then that these external controls are similar to the group of people who got treated in the trial so that the comparison itself is fair. So for that reason, the external control pool frequently starts out bigger than the pool of people in the trial. And first, some people may be eliminated because they're too different, such as if their ages are different, If the trial is, for example, in 4 to 7-year-olds, you wouldn't want to compare it to an external control pool full of 12 to 18-year-olds because that wouldn't be fair. So once you have a pool that's similar in its major demographics, then we use a statistical technique called propensity weighting that makes the control pool as similar as possible based on functional characteristics to the trial participants. So through a little bit of inclusion-exclusion and a little bit of statistics, you end up with an external control cohort that wasn't in the trial, but looks a lot like the patients that were in the trial when they first entered the trial. And so we then compare the performance of these external cohorts who were not treated compared to the open label or the open label people in the trials, or in the case of a placebo-controlled trial, after the time that the placebo arm ends. So for example, in the 102 trial, our placebo arm was only 48 weeks, but we'd still like to know how the patients who were treated do at years 2, 3, 4, and 5. But we won't have a placebo arm to directly compare, so we would use the external control past the part of the placebo. Okay, so I've given you a lot of trial development questions and logistical considerations, but let's talk about the results themselves. So first, let's look at Study 101. Study 101 was our first in human study, and so it's the smallest study we've done so far, with 4 patients that were aged 4 to 7 years old. All 4 of them received study drug, so this was an open label trial. The patients had a biopsy done before they received the study drug, and then again about 12 weeks or 90 days after they received the study drug. They've continued to be monitored in the trial for now over 4 years and will continue until they hit 5 years. And in particular, we've been having repeated measurements of the North Star ambulatory assessment, and that's what we'll be showing you today. So when we look at, there we go, when we look at their ambulatory assessments of the North Star ambulatory assessment, over 4 years, we can see here the performance of the 4 boys. As you can see in the bar graph, that's their total NSAA each year. So the purple is the first year before they got the drug. The blue is the first year at the end of the year in which they got the drug. And then the gray, green, and beige are years 2, 3, and 4 after study drug. As you can see in the table in the bottom, by those beige scores or the beige bars at the end, some of those boys are 10 years old, and they still have NSAA scores in the 20s. If you recall from my description of the NSAA, higher scores would indicate that people have kept their skills. So the fact that these patients had their scores increase means they maintained or gained skills or became better at skills than they had when they were first measured. I'm going to show you the same results in another way. So you saw bar graphs before. This time I'm going to show you on the left a line graph. In this line graph, the top bar, or the purple line that you're seeing, are the 4 boys who were treated. The blue line that you see below it is the external control pool that we talked about. So here I'm comparing how the treated boys did to how boys who never received 9001 did over the same span of time. You can see in the table below it that there were more patients in the external control cohort than there were in the treated cohort. So each year when we assess the treated boys, there are 4 boys, so you see a 4 in there. But you can see in the external control set, there were anywhere from 19 to 21 boys whose average created the line above there. So we feel pretty confident that that represents a reasonable sampling of boys over that period of time. The difference at year 4 is 9.9 points in the NSAA in terms of raw scores. On the right, you can see what happens if we model that using regression, which is a statistical technique. When we use regression, we can adjust for things like age or baseline functionality. And when we have those adjustments, you can see that the difference between the baseline and the 4-year score is 9.4. So 9.4 to 9.9 points implies that there are either 9 skills that got a little bit better or didn't get any worse, or maybe some skills were gained or their loss was prevented. Let's take a look now at the one-year update from cohort 103. Cohort 1 from study 103. So study 103 looked a lot like study 101 in that it was an open label study. So everybody in this study received the study drug. In this case, 103 has several different cohorts. Cohort 1 was ambulatory boys from 4 to 7 years old. There are several other cohorts that we will describe when I touch on the safety analysis, but they're not included in this because they haven't reached their 52-year mark. So this is the 52-week. It'd be amazing if it was 52 years. This is the 52-week functional data from those first 20 patients in cohort 1. They also had a biopsy before they received the study drug and afterwards, and annual assessments with the North Star. In this case, I don't have a line graph because we would only have the baseline and 52-week points. So I'm only going to show you the bar graph. And in this bar graph, we can see that on average, the 20 boys who received drug, who are shown in the purple column, had a 3.9 point increase in their total NSAA score. And the external control, so again, this was open label, so we didn't have any placebo patients in this trial. So the external control cohort on average, and there were 91 patients in the external control that led to this average, their total NSAA score went up by 0.8 points. This difference was considered significant by statistics, as demonstrated by the p-value that's up there. But what that hopefully means is that the boys in the treated cohort, we would hope, had some improvement in skills that they were able to do. 4 points is 1 point times 4 skills, or potentially 2 new skills if they went from a 0 to a 2. Along with the North Star, we do frequently get timed function tests as well. Not all of the timed function tests that we do in our trials have external data available, but 2 of them do. The time to rise from floor as well as the 10-meter walk run. These are also elements of the North Star, so they are commonly done. Time to rise from floor is the time it takes for a boy who's lying on the floor to get all the way to standing. So you can imagine that if the time goes down, that means that the boy was able to get up faster. Whereas the time goes up, it means that it took longer for a patient to get from the floor all the way to standing. And as you can see in the graph on the left, the boys who were treated with SRP-9001 had an average decrease of 0.5 seconds in their time to rise from floor compared to the external controls. Again, there were 91 that made up this average, had a slightly over 0.5 second increase in the time it took them to get up off the floor. Similarly, for the 10-meter walk run, this is a test to see how quickly a person can run 10 meters. So in the boys who were treated with SRP-9001, on average, they had an almost 0.8 second decrease in the time it took them to go that distance, compared to the external controls in the blue bar, who had a slight increase of about 0.2 seconds on average for how long it took them to move that 10 meters. Both of these were statistically significant, meaning that on average, there was considered a difference between these two, the treated groups who received 9001 and the boys who did not. So now let's look at an integrated safety analysis. So as I mentioned, we were able to pool the data from all 3 of the first 3 studies that we did. And as a reminder, that was study 101, study 102, and study 103. So you've seen some results from study 101 and the structure of that, and you saw the structure of study 103. I had told you that the functional results we showed were from cohort 1, which was 4 to 7-year-old ambulatory boys. As I mentioned, there are multiple cohorts in this study. So cohort 2 is older ambulatory boys, meaning that they were aged 8 to 17 years old, but still retained the ability to walk. Cohort 3 was any age and non-ambulatory, and cohort 4 was 3 years old and walking. In addition to these 2, I mentioned study 102 was our placebo-controlled trial. So study 102 involved 20 patients that in the beginning of the study received SRP-9001 and 21 boys who at the beginning received a placebo. And at the time, none of the investigators, the families, the boys, or anybody at Sarepta knew who got which one. After following them for 48 weeks, they then did what's called crossing over. The boys who received the placebo went on to receive SRP-9001, and the boys who had received SRP-9001 in the first half now got a placebo. In this case, the placebo was just some IV fluids. At the time that they crossed over, everybody still didn't know who got what, and we followed them for another 48 weeks. So at the end of both of those roughly 1-year periods, only then did we find out who was dosed with drug in part 1 and who was dosed with drug in part 2. At this point, we are over 2 years out from the beginning of the trial, so this study has been unblinded, and all 41 of the patients who entered the study have now received SRP-9001. However, as I mentioned, half of them got it a year ago and half of them got it 2 years ago. So this integrated analysis includes all of these patients and the safety experience that all of them had in the trials until the point of April 2022. As I mentioned, 85 people have actually been enrolled across all of these 3 trials, but at the time we did this safety cut, the very last, the 85th patient, was dosed just a couple of days after we pulled all of this data. As you can imagine, it takes some time for us to analyze it, and that's why not everyone has always been dosed when we pull these data. So the most common adverse events or sort of problems that came up in the trial are vomiting and decreased appetite. And these were reasonably common, as you can see. The number is the number of patients out of 84 that experienced it. And in the parentheses, you can see the percentage. So vomiting, for example, occurred in 52 out of 84 patients, which is about 62% of the patients who were dosed in the trials. I'll also explain what the 2 columns mean. As you can see in the middle, it says target dose, and on the right, it says all. That's because in 102, a number of patients got slightly smaller doses than the dose that we now use widely in all of our trials and would aim to have commercially approved. So the target dose is 72, so it's the 84 patients minus the 12 who received a slightly lower dose. But as you can see in the columns, the rates at which the adverse events happened was pretty similar between target dose and any dose. For the pain in extremity and procedural pain, I'll point out that many of these boys underwent biopsies, and so likely those were related to study procedures. There were also 2 significant events that I'd like to call out specifically, in addition to the common events that you saw on the previous slide. These are what we call serious adverse events, and these occurred in fewer patients. So vomiting is listed here for 2 patients because they had to go to the hospital and get IV fluids from the hospital. The liver injury and increased transaminases were people who were hospitalized or received extra doses of steroids or other interventions or investigations to understand what happened. All of those patients have recovered and without any ongoing problems. Rhabdomyolysis is an example of muscle breakdown, and that can happen in patients who do receive study drug, but it also happens in lots of patients who don't receive study drug. In this case, 2 patients required some IV fluids to help flush the proteins out of their system, and they also recovered quickly and without any long-term problems. The 2 I'll talk about in more depth are the ones that are in the colored boxes. So in the beigey green box is immune-mediated myositis, and I believe there's been a webinar on this before, so hopefully this isn't coming out of nowhere for anybody. But immune-mediated myositis means that there were muscle aches and weakness that were associated with the immune system reacting to the study drug. In this case, it was a 9-year-old boy that had a mutation that is now excluded from our trials. The mutation itself is considered part of the risk for this problem. And so this particular mutation would no longer qualify for Sarepta trials. but the patient himself experienced muscle weakness that also included difficulty in breathing and swallowing, because those are also activities that require muscular intervention. Notably, his heart muscle did not appear to be involved. He did receive a couple of different therapies, since we didn't fully understand the mechanism of what was going on. He received both plasmapheresis, which is a procedure that takes the blood plasma out of your body and gives you the blood cells back, and that works the best against antibody-mediated issues, as well as tacrolimus, which is a very powerful immunosuppressive drug. Eventually, he was discharged on just the tacrolimus without further plasmapheresis, and he has remained on the tacrolimus since then. He has gone home and he's able to breathe and eat by himself without any assistance, but he continues to take that extra drug, and so that is considered a sequelae or an ongoing part of this reaction. The other box in blue is myocarditis. So this is a heart issue that I don't think has been discussed extensively before here, although we did talk about it recently at a conference called ICNMD. So this was a boy who was 11 years old, and he initially came to the hospital for nausea and vomiting. And at that time, they checked a bunch of different blood enzymes and found that one of them, called troponin, was elevated. Troponin is a marker of myocardial injury, but can also signify a leaky heart muscle membrane. So troponin in the heart is a little bit equivalent to CK in the muscles. And I can imagine many people on this call are familiar with the test of CK, or creatinine kinase. So his troponin was elevated. They did do some heart tests to check that his heart was still functioning well, and his echocardiogram showed that the heart was squeezing normally. He had a cardiac MRI, which looks for something called late gadolinium enhancement. Late gadolinium enhancement is the pattern that shows up in Duchenne muscular dystrophy cardiomyopathy, but it's also the pattern that shows up when you have other forms of heart inflammation. And his cardiac MRI did show an increase in this pattern of inflammation compared to what he had had 1 year prior. He did receive some extra steroids, and he did go on some extra cardiac medications that he takes in tablet form every day, but was discharged from the hospital with normal heart function. And 4 months after being discharged, continues to have normal heart function. We are continuing to monitor both of these cases to see how they do in the longer term. So let's take a look at the functional data from these 3 studies. That was the safety data. Let's look at their functional data. So the number of people that we're talking about when we talk about the functional data is going to be a little bit fewer people than we saw when we looked at the safety data. So I mentioned that there were 85 enrolled, 84 that had data available when we pulled all this data in April, but not everybody had reached 52 weeks when we pulled all of this data in April. In particular, cohorts 2, 3, and 4 in study 103, because they started later, have not yet reached weeks 52, and therefore at the time that we took this data, we couldn't include them in the analysis. So the number of people whose 52-week functional data that we could put together in this pool is 52. And just like we did with the other studies, we used an external control for this. So as I'd mentioned with 101 and with 103, because everybody in this study got the drug, we need to use people that were not in the study as the comparisons. And we did a very similar type of comparison to what we showed you before. And here's the bar graph for that comparison. So the pooled group of 52 treated boys with SRP-9001 are in the purple bar, and on average, those 52 boys showed an average increase in NSAA score at 1 year of 2.3 points. And the external controls who did not get any form of treatment, they had on average a 0.1 decrease in the total score of NSAA. So pretty much stayed the same over the 52 weeks as opposed to the increase that we saw in the treated boys. and this was significant. Similarly, we got those timed function tests that I mentioned before. So on the left, you can see the time to rise from floor, and the 52 boys on average who were treated with SRP-9001 had a decrease in time of 0.53 seconds, meaning that on average, they got up from the floor about a half a second faster at 1 year than they had before they got the study drug, compared to the external control pool, who on average got up off the floor an average of 1 second slower at a year than they did at the beginning of the period. With the 10-meter walk-run, similarly, the boys treated with 9001 on average got a 10-meter distance faster, 2.23 seconds faster than they did at the beginning of the study. And for the external controls, it actually took them about a half a second longer to complete 10 meters than it did a year prior. So let's summarize everything that I just talked about. So today, what I've shown you is 4-year functional data from study 9001. So that's how those boys have done over 4 years in total, as well as integrated safety data from all 3 of the studies, 101, 102, and 103. We also showed you the 1-year functional data from cohort 1 of 103 as well. We're continuing to collect data in all 3 of these studies, as well as our other ongoing studies, the most notable of which, or the one you may have heard about, is EMBARK. So we are, of course, collecting data in that one as well. I'll also touch on future studies because I think those are the source of a lot of questions to Sarepta. So our future goals are to expand the eligibility criteria, as ultimately we'd like to make our drug available to everybody. And to that end, the most immediate steps we're taking are a new study called 303 Envision, which you may also have heard about at different points. And that will include 2 cohorts. One is individuals who are non-ambulatory with no age restriction, and a cohort of individuals who are 8 to 17 years old have retained ambulatory function. We're also planning to add a small cohort to study 103, which is the study with the 4 cohorts already that I mentioned in today's presentation. That additional cohort is hopefully going to explore dosing patients with mutations in exons 1 through 17, which is the region that has been excluded from EMBARK. That is going to be an extremely small cohort, and I'd like to emphasize that we will present more details when they are available, but given the size of that cohort, it is likely to be filled by patients that are already identified by the physicians that will be dosing in that study. And we will, of course, share more details on any future studies as they become available. And that includes studies about overcoming pre-existing antibodies, as I know that's also a major interest to the community and to Sarepta. So we hope to be able to give you updates about studies that address pre-existing antibodies in the future as well. I would like to say a particular thank you to PPMD for hosting us today in this webinar, to everybody who's here listening for taking time out of your schedule and having interest in the therapies that we are designing. But most strongly, I would like to extend Sarepta's thanks to all of the patients that participate in our trials, who are interested in our trials, and to all of the centers and their staff and their physicians who help us dose in our trials as well, as we wouldn't be able to move any of these therapies forward without this very large and very active community. So thank you to everybody.
Thank you, Stephanie. That was really very good. It's Pat Furlong here and really comprehensive. If you don't mind, we have many questions, as you might imagine, that have come in. And so I'd like to do this sort of systematically by going back through the slides. Is that acceptable to you?
Of course. Is there a slide you'd like me to go back to first?
Yeah, well, let's start at the beginning. Let's just start like around slide 4.
Okay.
And that you slide number 4. If we can go back there. Perfect. Okay. I think you mentioned that this is a global study and you have partners at Roche, and I know that they're probably listening and not actively presenting. But there are questions about when we talk about globally. Are we talking about India? Are we talking about Mexico, South America? Are we talking about really globally into areas that have not perhaps yet seen studies? Can you just describe that a little bit in terms of trials?
Yep. Hi, Pat. This is Louise. Maybe I'll start out and then I'll turn to
Great. Thank you.
Yeah. So we, as you know, as we've described, we partner with Roche because of their global reach, and that's both clinically, commercially, and in terms of the regulatory expertise. And so we have done our trials globally, and I'll have Stephanie comment on where we are with 301. But in terms of our regulatory pathway outside of the U.S., Roche is responsible for that, both the approvals and commercialization, and so they're going to be engaging with those regulators and they can discuss the registration outside of the U.S. But in terms of the clinical trials, Stephanie, maybe you want to touch on the global aspects of our current clinical trial.
Yes, sure. Thank you, Louise. So the 301 trial, which is EMBARK, that one is very near to completion, and at the moment we don't anticipate screening in any additional countries or any additional sites at this time. For the 303 Envision study, I think that is still in the planning stages. So a final list of countries and sites is not publishable at this time, but as with all of our studies, we will update ct.gov and the study website as new sites and regions come online.
Great, thank you for that. And I certainly understand that we will all, and I think many of the families are checking clinicaltrials.gov probably on a daily basis. I know how important this study is to our community. And just a couple more questions about this Envision study that is suggested to be in many, many sites. Will this be also restricted in terms of the inclusion criteria based on mutation? And within that mutation inclusion criteria, 18 to 58, that you're talking about, do you also include duplications within that range as well?
That's a great question. And we currently do expect that EMBARK will have a restriction over exons 1 to 17, and I would emphasize that that is because of that safety episode that I described. We want to ensure that we balance getting the drug to everyone, which is of course our desire and our hope, while also making sure that we aren't causing any harm or undue risk, and that we can dose everybody in a safe way as well as an effective way. And so that's why we want to do additional studies, such as the cohort I outlined for 103, in order to assess how we can approach those mutations in the most safe way possible. So until we have a little bit more data, I would expect that the Envision study will carry the same exclusion. Duplication mutations have been allowed in our studies since the inception. It's usually about whether or not there is a shift in the reading frame. But duplications were allowed in EMBARK as well, for example.
Right, that's what I thought. I just wanted to make sure that that was clear, as long as those duplications were within the range of inclusion in terms of mutation specific for the studies themselves. So in the study that you talked about, I may have missed it. How many people are you recruiting to that Envision study once it opens? Are you able to tell us numbers, or is that still in development?
It's still in development. We do anticipate that the total size of the study is probably more similar to the EMBARK study than it is to the cohorts in 103, for example. Those are very, very small cohorts. But the final numbers will be released when the study goes live on ct.gov.
Great. And you mentioned that EMBARK, which is the phase 3 study that is now in process and nearly complete, as you mentioned. Can you tell us how many were involved in EMBARK, or are involved in EMBARK?
So the target enrollment for EMBARK was 120 patients. So that that remains the target for total patient enrollment is 120.
Great, thank you. And you're nearing completion of that enrollment, is that correct?
Correct.
Great. All right, and one last question. When you mentioned the study that you will be looking at the mutations that have been previously excluded, 1 to 17, and this is a very small study, as you mentioned, do you anticipate this will be specifically in U.S. or ex-U.S.? You already mentioned that the patients have likely been identified, but just for our, as a community understanding, is this really limited to the U.S. Or is it going to be outside the U.S.?
So I would anticipate that cohort in 103 will be U.S. The 103 study itself, all of the sites that so far participate in that study are in the U.S. And so I would anticipate that we do not anticipate expanding the trial to ex-U.S. for this cohort.
Great, thank you. That's very helpful. So can we go to slide 5 and look at some studies? I think, Stephanie, you talked about study 102, and I'd like to have a little more disclosure on 102. 41 patients in that study. You mentioned in a later slide, and I think that I have that slide where the dosing might have been different. You had a target dose, and you mentioned that there was some difference in dosing. Can you talk to us about study 102, the results of study 102? Are you able to talk about them? As you've talked about 101 and 103, can you give us a little more, shed some light on 102 and what happened there?
Sure. So 102 was, as I mentioned, a double-blind, placebo-controlled randomized trial. And so as I mentioned, half the patients were dosed at the beginning of the trial and half the patients were dosed at the crossover period at the end of year 1. When we began the trial, everybody was actually intended to receive the same dose, and the way that we assess the number of genomes in the study drug, the standard for that changed by the external sort of scientific community created a new way of measuring that while we were doing the study, and when we applied this new standard, it gave us slightly different results for how those lots were composed. And so that meant that under the new standard, there were slightly different doses between the different lots that were used in that study. As a result, then, there is more than one dose when we calculate the study. So there were 12 patients who received a slightly different dose than the intended target dose. And so that's why on one of the later slides, in the safety slide, I believe, that I showed towards the end of the presentation, slide 18, we had separated out this concept of target dose versus the other doses. In terms of the results of 102, I would point out that in the placebo-controlled portion in year 1, and I believe we've discussed those results in a previous webinar as well. So if people are interested in that study, I don't know if that recording is available somewhere as well. But in the first year of that study, there was a significant difference between the treated group that were 4 and 5-year-olds, between the placebo and the treated group. In the 6 to 7-year-olds, we found, unfortunately, that the patients in the placebo group were, in fact, not well matched to the patients that were in the treated group. So the placebo group patients had much higher functional scores at the start of the study than the treated group did. And so it became very hard to make accurate comparisons of how they did over time. And so they weren't statistically significant. However, we did this same kind of external control analysis that I've been showing today in 102, and that did show that there was a significant difference between what happens to patients when they're not treated versus the 6 to 7-year-olds that were treated. But as I said, I think a more complete explanation of this was all done in a previous presentation, so I would encourage people to look for that as well.
Yeah, I know there's a lot of interest in that study and trying to sort of sort through and understand what happened, what the results were. I do recognize that in some, there was a dose, potentially different dose, or different in terms of, I guess if you wouldn't mind, different in terms of lower dose or different in terms of the same dose but calculated on a different metric. Can you sort of sort through that so we understand it a little bit better?
Of course. So it is truthfully a lower dose. So the method for measuring became more precise. As I said, the scientific community, the technology got updated. And so when we went back and looked at those doses retrospectively using the new technique, we were able to get a more precise measurement. And so some of the doses were lower than they were in the intended dose. Now, all of our doses since then, of course, have used the most current technical standard and therefore are all that sort of what we call target dose.
Right. So it's a much more stable measurement in other studies. So I guess then I think what the patients and families would probably want to know is, and I think all through these studies, as you know, the real question from any single family is how did my son do or how did I do as a patient and participating in the study? Is Sarepta planning to release individual results to include biopsy, North Star, and other functional assessments so that people can understand where their child began at baseline and what the impact of the gene therapy was on their particular child?
That's a great question, and I've certainly heard that people are interested. Trial participants, of course, are very interested to know their own results, and so I can understand that they would want to hear their own results. That's something that we're sort of talking about internally as a company about how to do that and provide appropriate context to what those results mean and all of that kind of thing. So I don't think I can definitively make a promise to the community that we will definitively give individual results, but I would also refer to Louise to comment on this.
Yeah.
Thanks, Stephanie.
No, thank you.
Louise?
Yeah, so the results will be disseminated in a publication, but then also the study is currently ongoing, and so at the end of the study, the results will be shared, and then each of the participants should discuss with their study doctor for individual results. But the study is still ongoing at this point, so once the study is complete, we'll share.
Yeah, thanks, Louise. That's really helpful. So and I apologize for all these detailed questions. So what we're talking about then is at the completion of the study and the 5-year mark, or at what point in time would those results likely be available? Excuse me. We're all very familiar with this 5-year. The FDA requires a 5-year view, basically, of the safety of these studies. And so in Sarepta's mind about potentially giving individual results to the study physicians to disseminate to the families, are you looking at when the study is completely closed, 102, in terms of functional measurements 1 year after the last patient in, or are you talking about the 5-year mark that FDA is often referring to?
Yeah. It's at the completion of the study when we have what's called the CSR, the complete study report, is when that. So certainly I think each patient that participates in the study should discuss that with their doctor.
Great. Thank you. Thank you for that, and thank you for clearing that up. Could we go to slide 11?
Of course.
Back up one to slide 10 on those first 4 boys. First of all, I think this is remarkable, especially when you look at where these young boys were at baseline and then the improvement that they received. I think we're seeing a stabilizing or in some, a very slight decline. This brings us up to the issue of redosing. How do you think about it? What is your timeline for it? I know that Sarepta is deeply involved in this effort. So can you shed a little light on the idea or the thought or the timeline for redosing?
Yeah, I'm happy to take that. So for redosing, obviously, we're very pleased with the durability that we've seen in the clinical trials so far, as well as the preclinical data. We've, as you well know, Pat, we've undertaken several different ways of looking at treating patients with those pre-existing antibodies and then the potential for redosing if it's needed. So we currently have internal studies looking at apheresis. We have external partnerships looking at other ways to remove antibodies. So those studies are currently in the research phase, and as soon as those progress to the clinical stage, we'll let the community know. But obviously, we would be focused on the patients with pre-existing antibodies first, and then if redosing is needed, that would obviously come next, and we would visit that clinically first.
Right, thank you. And that was going to be my next question that you've already answered. Thank you, Louise, about people who are excluded based on a threshold of antibodies. So you're already working on utilizing both internal and external sources to think about what ways you could employ that would decrease the circulating antibodies if someone was over the threshold. And that would be your first step, followed by another step at another point in time of trying to decrease the antibodies of someone who's been in a gene therapy protocol to be able to redose them. Do I have that right?
That's correct. Thank you. Perfect.
Okay, great. In the Endeavor study, can we go to slide, I think it's 13, where you talked about the Endeavor study. And you also talked about part of this Endeavor study is going to be recruiting littles, as I call them, so below the age of 4. Can you talk to us about the timeline for that? When will it start? How are you thinking about it? How many would you be thinking about including? Can you talk to us a little bit about how you're thinking of treating those little guys?
Sure. So the cohort 4 of this study was all 3-year-olds. So we have dosed 7 3-year-olds already as cohort 4 of study 103. We don't currently have plans within the 103 study to dose any patients younger than the age of 3, although I believe our Roche partners are working on developing a study that would go younger than the age of 3 as well, and so that may be a future study.
Great, thank you for that. We're, as you might imagine, the enthusiasm about treating as early as possible to preserve muscle function is front and center on all of our minds, so we thank you for that. If you'll go to slide 18.
Sure.
So you talked about vomiting. Do you see this? I know that in other studies that have been approved, such as Zolgensma, we've seen vomiting. And do you see that as a sort of a standard? You've got a percentage of children who are vomiting shortly after the infusion. I could assume that maybe their parents are too, with the anxiety around a child in a gene therapy study. But do you see this as fairly common, and in your knowledge and experience, exploration of other studies, is that something we see pretty frequently?
It's certainly been our experience that in every study that we've run, we have seen that vomiting seems to be, as you can see, a fairly common adverse event. It does tend to be, of course, not very serious. Most of these children don't have to go to the hospital, don't need any IV fluids or anything like that. It's I'm certain not fun for the patients or their families, but it doesn't seem to be medically serious as a side effect. I agree with what you say, that it's certainly been seen in other gene therapy trials as well, but I think we're just glad that the most common adverse effect doesn't seem to be a particularly serious one.
And Stephanie, if I remember correctly, it's pretty short-lived. It doesn't last for days and days. It's usually a 48-hour plus or minus experience, which doesn't make vomiting easier, but at least there's an endpoint to it.
Correct. It seems to be limited to the first week after therapy. Of course, there's going to be a range. Some kids don't have any, and some will have it last a little bit longer. But in general, I'd say for most of the children who experience this, it's quite short-lived.
Great, thank you. And let's go to slide 19. So Stephanie, you mentioned these two serious adverse events in terms of the child who experienced the immune-mediated myositis, the 9-year-old, and then the 11-year-old. A couple questions around this. First of all, these two experiences really made you, that experience created the need for, or in the interest of safety, to modify the protocol to exclude certain exons. Is that right?
Yes. So the event in the greenish beige box, the immune-mediated myositis, is in fact, as you say, the event that led to the exclusion of exons 1 through 17 in the phase 3 trials, EMBARK, and will likely be true in ENVISION as well, because of course this was a serious event. The child was in the hospital for a couple of weeks, and so we want to be sure that we can dose safely and with benefit to anybody that has a mutation that increases the risk for this kind of event.
Right. And the young boy with myocarditis, do you feel that was directly related to gene therapy, or can you give us a little more detail about, in addition to the additional cardiac meds and the increase in prednisone, do you think that was a response to the transgene, or how do you think that was related to the study?
So I don't think it was related to the transgene. So the transgene itself would not be expressed in the body yet at the time at which this occurred. So this occurred very soon after dosing, and the transgene would not yet be expressed. So I don't think this was a reaction to the transgene the way that the myositis was. In the case of the myocarditis, there's only been one case, and it's also a very similar type of case has been described in the literature in patients who have not been exposed to gene therapy. So there are cases reported where something similar happens, where there's a very high troponin level, which is that marker that I was talking about. It's a very high troponin level, and then a change in the cardiac MRI, and those patients have not been exposed to any kind of gene therapy. So with only one case, it's very hard to know for certain whether this is related to the underlying Duchenne cardiomyopathy phenomenon that can occur, or whether this was precipitated by the gene therapy. I think out of an abundance of caution at the moment, we certainly wouldn't want to rule out the possibility that this is related to the gene therapy, especially given how soon after dosing it occurred. And so that's why we're taking this very seriously and assuming for now that the two are related. Certainly the investigator marked them as being related, and we don't disagree with that, but I think it's too early with one event for us to fully understand completely what is happening with this event, and that's also why our trials, all of them, monitor troponin levels, which is again, that marker that I was talking about, so that we can keep an eye out for any indication that there is a true safety signal occurring.
Yeah, thank you for that, Stephanie. I think that we've seen in Duchenne, certainly in speaking to the cardiologist, that we do see these increase in troponin levels, and sometimes accompanied by a decrease in the ejection fraction in Duchenne, not necessarily within a gene therapy, obviously not in a gene therapy study, or I should say outside of a gene therapy study. We've been working with the Action Network, as you know, really to begin to better understand the heart and address some of the cardiac issues from a natural history standpoint and from intervention with new medicine. So I appreciate the explanation on this. As we think about how you've modified your protocol based on the event, the immune-mediated myositis, there are questions still about what kinds of mutations are included. Duplications you've already answered. Also, point mutations, as long as they're within the inclusion criteria, would also be included in these studies and not excluded. Is that correct?
That's correct. So I would say that currently for the EMBARK study, and I think this is actually on our ct.gov page now as well, that the genetic inclusion criteria require a frame shift or a nonsense mutation. That shift is, that causes a premature stop, a nonsense mutation, and it can be a frame shift deletion or duplication. I think we also have accepted splice site variants before. So this is something that I think anybody that has a question about what these inclusion criteria mean and what the genetics mean should have a discussion with, their physician about what exactly their mutation is and how that lines up with the different inclusion criteria. And if their physician has a question, they can always reach out to us as well, because the genetics can be quite complicated with all the different types of mutations and locations of mutations and things like that.
Right. Thank you for that. I know that you publicly announced your intention to submit to the regulatory agency in the United States, the Center for Biologics, with the hope and with certainly the promising data that you have and the desire to get an approval so that more patients can have access to gene therapy. Can you give us a little bit of your timeline about that? And is there, and I know Rosh is not on the line as well, but are you able to at least say there's a similar activity going on with the EMA, or can you give us a little bit of color about the regulatory next steps?
Yeah, thanks, Pat, for that question. We're certainly very excited about submitting for accelerated approval, and so as we've commented, we'll submit the license application in the U.S. this fall. That will be followed by regulatory activities extending through the spring. Certainly, Roche is in 100% partnership with us and following similar strategies. Their base case is the EMBARK trial, the 301 trial, in terms of that. But certainly, as we are all trying to move as quickly as possible, we are looking for avenues, but certainly the base case ex-US is 301.
Great. Thank you for that, Louise. I think, as you know, there are a growing number, as you've just stated, of people who've been in a gene therapy protocol with Sarepta. We're all very enthusiastic and continue to be hopeful for an approval in this country and certainly many other countries. One of the questions that we all have as parents when our children receive a therapy, whatever that therapy is, and in this case a gene therapy, is how do we think about next steps? What happens? How do we think about combinations? For example, if an individual was amenable to one of your antisense oligonucleotides that's been approved, can we combine those therapies? How do you see this going forward so that we can get to a terrific combination that really halts disease progression or ends Duchenne in terms of progression? Can you speak to that idea, or what you would need to know, what doctors would need to know in order to think about combination therapies?
It's a great question. It's something that we think about a lot. So we have ongoing research studies that are looking at combination therapy. Certainly there's no, in data we have to date, no contraindication, meaning that there's been no problems identifying with receiving both an exon skipping drug and gene therapy. Certainly when we talk about combination, you could be talking about receiving exon skipping first and then gene therapy, or vice versa. And so looking at both of those strategies. And so what we're doing now is determining whether clinical trial is needed or if this is just real-world data that can be collected. Certainly, we have treated patients that were previously treated with gene therapy that were previously treated with exon skipping already. And so we're collecting the totality of that data, and we'll present it when it's ready. But certainly something that we look at, as you can imagine, as a multi-platform company, quite a lot.
Yes, I'm happy it's on your radar. Louise, I have a question for you, and this you can tell me that we have no idea about this, but I've always wondered, when we get to the point, and I believe we will, of redosing these patients, do you believe in, as you've done so much work on the basic science of this and working with Jerry Mendell earlier and now doing all the work with Sarepta, do you expect or anticipate that a redosing strategy would be cumulative, would be complementary? How do you see that going forward in terms of what could we expect?
In terms of redosing just gene therapy? Sorry, I just want to make sure I'm answering the correct question.
You redose a patient who's had gene therapy. What would you expect? It would sort of be a boost to what they've already had. Do you see any issues with it scientifically? Do you feel that it would be complementary to what they've had or how do you see that biologically?
It's a difficult question to answer. I'd say one of the challenges with redosing is the level of antibodies that a patient that receives gene therapy generates. So they're much higher than the person really occurring antibodies, and so getting them low enough. What we don't know is getting them down to a safe level, just making sure if there's any unanticipated problems with redosing a patient that we're currently. We have done large animal studies, and those have gone well so far. I would anticipate that maybe you wouldn't get double, for example. Maybe it would be something less than that, but we honestly just don't know yet, but we are working hard at that.
Yeah, I figured you didn't know, but I thought I'd throw it out there because as we talk about redosing and hope for redosing, I just wondered how you feel about that. Well, first of all, I'm going to turn this back over to Eric, but Stephanie and Louise, I can't thank you enough. We certainly wish for all the best and the best of luck going through the regulatory process because it matters to so many of us that are listening today and so many of us that will watch this at some later point and are anxiously awaiting a gene therapy. So thank you for all you do, and I'll send this back to Eric.
Thank you, Pat. I actually think in your discussion, you tackled most of the questions that we saw come through from the community. So thank you both, Stephanie and Louise, for answering all of those questions. This is just a reminder to everyone, this is a recorded webinar and it will be posted to PPMD's website, so please feel free to share when that is uploaded. But Stephanie, Louise, thank you again for joining us to talk through some of the results from these initial studies, and we look forward to future updates from you.